to switch things around a little bit today. As you guys noticed from the quiz, there are 2 different stains that we're going to do today that require air drying. Now we could go ahead and I'll teach you about staining, and then you can start the lab. And the air drying will take you anywhere from 30 minutes to an hour. You just have to sit there and wait until it dries. Or we can do those 2 first. So we're going to do those 2 first so that while I'm talking about staining, they're drying, and then you don't have to wait later. So what I want you to do, you guys each have a slide dispenser in your trays. I'm going to borrow y'all's. It looks like this. To get a slide out, you press this little button, and it should shoot one out for you, just like that. Okay, you each need 4 slides. So go ahead and grab 4 slides out. And then while y'all are grabbing those out, I'm going to pass around the box of China markers. So those are my, have we used these yet in here? Yes. The fancy pencils. You'll need one of these each. So go ahead and pass that around. So you should have 4 slides each. Sometimes they stick together, so make sure you only have 4 and you don't have 2 that are stuck together. That'll make it hard to see under the scope. Okay, so on those slides, once you have 4, I want you to see if you can figure out which side of the slide is the top and which side is the bottom. Now, the top, you have one end of the slide that's frosted, so it looks white. The top of the slide, where it's white, if you reflect light in it, it's not going to reflect on the top of the slide. So the bottom of the slide has like some glass over that frosted area. You can also sometimes tell if you take your finger across it, the texture changes on the top. On the bottom, it's all glass over the bottom, so the texture doesn't change. Can y'all tell what I'm talking about? So find the top. The top is the side that's not shiny when you shine light over, or like hold it up to the light on the frosted end. If you're unsure, you can ask me. Okay, once you have those pencils, I want you to write on the frosted side, SI on one of them. And exactly like that, capital S, lowercase I. CA on one of them. And we'll do NE on one of them. And then the 4th one you won't label. So use that wax pencil for this, not a Sharpie or pen or pencil. Use one of the wax pencils for it. It doesn't matter what color. And then for the SI slide, I want you to draw a circle with the wax pencil that's about the size of a quarter or a nickel, like in between the 2, okay? Right in the middle of the slide. And I want you to do the same thing for the slide labeled CA, but not for the NE slide. Y'all with me so far? Okay, go ahead and turn on your micro incinerators so they're heating up.
You say we're running this on the frosted side?
Write the letters on the frosted side, but draw the circles in the middle of the slide, so they'll be on the glass, right in the middle. So if you have your frosted side labeled, it'll look like this. Does that help? About the size of a quarter should be good. Or nickel, that'll work too. Okay, so how you're going to do this, make sure your micro incinerators are turned on. Plug them in and turn them on so that they're heating up. We're going to set up our slides. Which two need to be air dried? Simple and capsule. So SI is for simple, CA is for capsule. The reason that I'm having you write it with the lowercase I and the lowercase a is because later on, when you're looking at these slides under the microscopes, if you accidentally have your slide on the scope upside down, you aren't going to see anything on the 100. You'll see it on the 4X, you'll see it on the 10X, you'll see it on the 40X, and then on the 100 it's going to disappear. So I can real quickly look at the side of your slide and see how it's labeled. If that's upside down, I'll be able to tell. So make sure you're working with it right side up the whole time. Okay, so we're going to set up the simple and the capsule. Your simple stain, and you know what? I might do this a little bit differently. Sorry guys, go ahead and set this slide aside, okay? Set your capsule slide aside. Grab another slide out. So you all need one more. Sorry guys. And don't draw a circle on it this time. I'm gonna do it different. This will be better. So just get a capsule slide out and label it CA. Don't use the other one. Okay, so for your simple, what are we going to use on that one? So you can look at your pre-lab for this. We're going to use water, and then what organism are we going to use? Bacillus megaterium. So what you're going to do is take your loop. You guys have water in your trays. You're going to use your micro incinerator. Take some water. Drop a drop of water onto your loop. So you're going to put your loop in the micro incinerator first, and then take a drop of water from the container in your tray. And after your loop has cooled, you'll just drop a drop on the end of the loop. And it may take you a minute to get a drop. If it doesn't want to do it for you, which this is looking like it's not going to want to do it, as long as you flamed your loop or stuck it in the micro incinerator, you can stick it in the water itself. So just take the dropper out and stick it in the water. Or what you could do, and what I recommend you do, take your dropper and drop a drop of water with your dropper, because it has a dropper, and drop it right into that circle that you drew. Does that make sense? Y'all with me? Okay. If the water touches that line or the circle that you drew, that's okay. You'll see the wax when you look at it under the microscope, but you'll know that it looks different. So you're going to drop water into the middle circle, and then you're going to micro incinerate your loop, and you're going to get Bacillus megaterium. So we have it over here. This Bacillus, you don't need much of it. Just tap your loop down and then mix it in the water. Okay, so you're going to tap your loop down onto the Bacillus and mix it in the water with your loop. Just mix it around in that little circle. And again, if some of the wax gets into the water, that's okay. Then we're going to let it dry. Bacillus megaterium. Okay, for your capsule stain, this one's a little bit different, okay? We're gonna set it up a similar way. Did you notice anything special about this one when you were reading? We have to use a spreader. What that means, you're gonna use another slide. That's it. So a spreader is just another slide. That's why I had you guys grab 4. Let's see if I can get this pulled up. What stains are we going to use for this one? India ink and crystal violet. So for our first step where we want it to air dry, we're going to use India ink. So the first thing we're going to drop on the slide is India ink. And you guys, this is not easy. I'll be honest with you. If you don't get it perfect the first time, that's fine. I've been doing this for years and it's still not perfect, okay? It's not the easiest thing in the world. So you already figured out which side is the top of the slide and you have it labeled, right? So it's labeled CA. You're gonna take India ink. This is in the box that you guys have, so you should have 1 box per pair. I hope you made sure everything was in there, because that'll be part of your work ethic. Your India ink looks like a dropper with a cap on it. That is not what it is. Do not try to take the cap off. So that's built in. You can take it off if you pull hard enough, and then India ink might go everywhere and stain a lot of things. Okay, so don't try and take a lid off of the India ink. Now, if you look at it, all of yours are new because you're the first lab section of the week. It's closed right now, or closed. It hasn't been used yet. How you use this the first time is a little weird. Where it says India ink on the little bottle, you have to squeeze. There's a little piece in there that's going to break, and then it will release the India ink up into the top so that you can drop it. So you just squeeze it. You'll hear it crack. And then when you flip it upside down, the India ink is going to come down. Y'all see what I'm talking about? Okay. Yeah, you have to squeeze pretty hard. Don't be gentle. Okay? So what you're going to do, and I'm going to show you guys this before you do it, because this is tricky. You're going to drop this on the end of the slide. So instead of in the middle, we're going to put it on the end. Just 1 drop. Just like that, okay? You're going to take your loop, put it in the micro incinerator, let it cool. What organism are you going to get? Bacillus megaterium for the capsule stain. You're going to mix it into the India ink at the end, okay? So mix it really well, and then you're going to take a spreader slide. It doesn't matter which side of the slide you use. How I normally do this, I'll hold the spreader slide with my dominant hand, and I'll hold down this slide with the India ink and the bacteria on it with my non-dominant hand. I'll drag from the frosted end back towards the India ink, and then you spread it forward. Okay, now you don't want to drag this all the way into that drop. You only want to drag it until you see India ink along the edge of this spreader slide. Okay, so I'll show you how I do it. It's easier if you do it quick, and the best result is normally about a 45-degree angle, if you can, maybe a teeny bit less. But this is how I do it. So I'm not going to pick up that whole drop with the end of my slide because that's going to be too much. So I'm going to drag this slide back until I see it hit the India ink. Do y'all see how it spread across my slide? And then push it forward. Okay, now some of yours, the India ink is going to go all the way across. It probably should look more like that. So again, this is not perfect. I wouldn't do it again, no. Because then it'll make this area there will be too much ink right here. So this is the perfect amount, and this will be fine. When you're looking at it under the microscope, you'll just look at an area that is black. Okay, and then we have to let this air dry. Okay, so it shouldn't take too long, but over here you're gonna have a bigger drop that's gonna take longer to dry. And you want the whole thing dry, so we're gonna do it now. This spreader slide is gonna go up to the front in this container. Okay, do you guys have any questions on this? Okay, I want you to wear gloves with India ink. I want you to wear goggles as well, so I'll get those out for you guys, just in case. If I were you, I would grab a paper towel and set your slides on a paper towel to dry and set them aside, because we're going to get out our notebooks in a minute. So you want them set aside so that they're not going to get hit or something like that while they're wet. Okay, go ahead and get started on that. While those are drying, we're going to move on here. So you'll want your lab manuals out. A little bit of business, I guess. Your midterms. Average was an 84, which is good. The high, I had several people get a 50 out of 50 or a 100, which is good. That's awesome. So if you want to see your midterms, schedule an appointment with me, and I'll show it to you. I've already had several people come by. Did you guys have any questions from your midterm? Like any specific ones you're dying to know the answer to? I know that was a long time ago, so. Okay, your final is going to be, I don't like the word cumulative for this scenario, but the stuff that we're gonna be going over the second half is building on the stuff we've already gone over. So you have to know all that. Does that make sense? Like when you're studying for the final, if there's something you find in the first half that we haven't really talked about at all in the second half, I wouldn't stress so much about going over that. Okay? But I could ask you anything from the lab manual on the final. Okay, so we're going to talk about staining. Hopefully those will be dry by the time we need to stain them. Did any of you guys notice your simple stain didn't spread out super easily? If it didn't spread out super easily, that could be because the slides sometimes have a film on them. In your lab manuals, you'll see it. It says to clean your microscope slides before you make them. I don't know if you guys noticed that in your lab manuals. And we're going to do that for the negative stain, which is that last slide that you have. But in the future, I think it's going to be really important to make sure you clean all of your slides. So in your little boxes, you have something called greased lightning, and it'll just remove the film off the slide. So what I would do in the future, and I'll remind you of this, just put a drop of it on both sides of the slide and wipe it off with a Kim wipe. That's it. And it'll prevent any of that, I guess, beading up if it looks like it was beading up a little bit, or if it wasn't spreading out very well for you. Yeah.
So for that one, we're supposed to put the drop of water and like mix it around.
Right. Yeah. But in the future, we'll use grease lightning for all of the slides to clean them. Okay, stains. If you guys looked at bacteria under the microscope, under our bright field microscopes, what color would it be? If you looked at bacteria under our bright field microscopes, what color would it be? Light, clear. Under the bright field, yeah.
If it's stained or not stained,
If it's not stained,
then whatever color is not the same.
what color is bacteria? It's normally like a translucent color. So if you don't stain it, you're not going to see it. You might, I should say, you might be able to see an outline if you look really hard and your microscope is adjusted perfectly. But that's not always the case. So that's why we're going to stain it. The problem with staining, though, is that it can distort the cells a little bit. So your rod-shaped organisms or bacillus-shaped organisms, picture perfect, would look kind of like that. But when you stain it, it might look more like that, because stain can distort the organism's shape a little bit. But this right here looks a lot more like a bacillus shape than a circle, right? So you should be able to tell the difference between a bacillus and a cocci shape. At this point, you've talked about that in lecture, right? The different shapes and morphology. So you have some negatives to it. We also have to expose the bacteria to heat. And I'll go through that as well when we're staining to make sure that it's killed. That heat also can distort it a little bit. So there are some negatives. However, the big thing is that stain creates contrast. I hope you remember that from when we talked about it before. So if we increase the contrast, what happens to the resolution? It gets better, right? So when we use a stain or just a pigmented substance, we're adding contrast, or the contrast is going to be higher. So today with the simple stain, you have bacillus on your slides right now. It's going to be translucent if you look at it right now. We're going to add a stain. Do you remember the stain that we're going to add? Methylene blue. When we add that stain, that bacteria is going to be a dark, deep blue. That's going to be a lot easier to see, a lot higher contrast than if we didn't stain it. Okay, so you have basic stains and you have acidic stains. That's listed in your lab manual. I'm going to give you a little bit more detail on this, and I want you to know this, okay? You may want to write it somewhere next to the spot in your lab manual where it has it. I believe it's on the bottom of 46. There's a beautiful space there for you to write this. Y'all see what I'm talking about? Okay, so stains can be basic and stains can be acidic. Bacteria, and you just have to memorize this, bacteria carry a negative charge, okay? If you know that, these will make a lot more sense to you. The basic and acidic will make more sense. And to be honest with you, it's not going to make a whole lot of sense this week, but you're definitely going to want to know it for the stains that we do next week. So this isn't our only staining lab. In fact, we're going to be staining pretty much every lab till the end of the semester. So knowing this is going to help you in the future. Okay, so we have basic stains, and I'm going to draw, I guess, a little chart here, and acidic stains. Basic stains have a positive charge. What do you think acidic stains have? A negative charge. So if bacteria has a negative charge, whoops, I forgot to write that up here. If bacteria carry a negative charge, do you think a basic stain with a positive charge will end up staining bacteria? If you remember from chemistry. Like opposites attract, right? Maybe sounding familiar a little bit. So a basic stain can stain bacteria. So I'll put stains the cell. An acidic stain has a negative charge. A negative charge is not going to be able to stain another thing that carries a negative charge. So an acidic stain is not going to stain our bacterial cells. So if we used an acidic stain, your bacterial cells are not being stained by it. It stains the background. So everything behind or beside the bacterial cells is going to be stained. So I call that the background. Stains the background. So I mentioned earlier we're going to stain our bacillus with a certain stain. Do you remember which one? Methylene blue is going to turn our bacterial cells a deep blue. So methylene blue is going to be what, basic or acidic? Basic. So an example here is going to be methylene blue. What's the other stain you've used today already? India ink. So India ink actually is not staining your bacillus right now. So you're letting it dry. It's staining everything but the bacillus. So it's staining the background, which makes it basic or acidic? Acidic. So an example of acidic would be India ink. Now for your capsule stain, you're using two different stains, remember? You used India ink already. What's the other one? Crystal violet is the other one. Crystal violet is a basic stain. So you could write that here if you want to. It's a basic stain, so it's going to stain the bacteria. What color do you think it'll be? Purple. So you're going to let the India ink dry all the way. Then you're going to put crystal violet on it, and it's going to turn purple. So the bacteria will be purple, and the background is going to be black. Y'all with me? Okay. Oh, one more thing, just for fun here. Do you guys know the chemistry term for positive charge? I'll just write this in. I think I heard somebody say it. Cationic for positive. So you might see it that way. What about anionic? That would be negative, right? Okay. In case you see that in your textbooks or something, a cationic stain would be a basic stain. Okay, so we're going to go through these stains that we're going to do today. You have three. The first one is the simple stain. I'll be honest with you, the simple stain is not done in the real world very often at all. So this is a picture of a simple stain. This is Streptococcus stained with crystal violet. Okay, so all they did was took Streptococcus, put it on a slide, and then stained it with crystal violet. And it turned purple so that you could see it under the scope. This is Bacillus megaterium with a simple stain. They used crystal violet as well. You guys are using Bacillus megaterium. What stain? Methylene blue. So yours will look like this, but blue. This is what yours will look like. If you did not just tap down on the bacillus, instead you scooped across the agar or something like that, it would look more like this. Do you see how many there are here? It's like a bunch of bacteria compared to this one where you can see each individual cell. So less is actually better in this scenario. Okay, so the simple stain, again, not really clinically significant. They do use it sometimes with fungi because with those you can tell based on the shape right away what the organism could be with most fungi. Here, all you can really tell is the morphology or the shape, and you can sort of tell the quantity too. So if you're only seeing one cell in a field from a patient sample, that's not very much, versus if a patient sample looked like this, that would be a lot. In your case, your patient sample I grew for you, so it's a little bit different. But if you could do a stain like this with, say, like a drop of urine from a patient and see if there's bacteria in it that way to check for a UTI. So you guys are going to do this one today. Your wet mount, which we haven't talked about at all yet, does it have an air drying step? No, so we haven't set that one up. This one, it's a little bit different. Staining typically kills the organisms that you're working with. Also, there's a step, which I haven't talked about yet, where we have to heat fix them, heating them, which I mentioned does distort the cells a little bit. However, it also kills them. With this wet mount, you're not going to be killing the organisms. They use it for viewing live organisms sometimes in the lab, or if they need to look at something really quickly, they'll use this one. Typically, this one's done on every spinal fluid that comes down into the lab. So if you ever draw spinal fluid on a patient, they're going to get a gram stain, which we're going to talk about next week, and they're going to get a wet mount. They're also going to get that direct microscopic count, if you remember me talking about that. So there are several tests that they're going to run on every spinal fluid. This is one of them, because there's one organism that has a very distinct look. It looks just like this under the microscope. It's called Cryptococcus neoformans, and they can diagnose it as soon as they see it on the microscope from a wet mount. This is not actually bacteria. It's a type of fungus, but you don't see it too often. Actually, I would say it's pretty rare. I think I've seen it maybe like twice. It's normally in immune-compromised patients, but it's done. They look for it for every patient that gets a spinal fluid taken from them. What you're going to do when you do this later, you're going to get India ink. So India ink is black. It stains the background. You're going to put water, and then you're going to put your organism, which for us, it's going to be Saccharomyces cerevisiae. We're not going to use Cryptococcus. It's a little dangerous. So I don't want to have it in here. It looks the exact same. So Saccharomyces cerevisiae is baker's yeast. But you're never going to see baker's yeast in somebody's spinal fluid. But you could see this, and it looks the exact same. So when you're looking at it under the microscope, this one's a little bit weird. This is what it's going to look like on the 4X. So when you're looking at it, these spots down here are bubbles. So if you see a big spot like this, they're bubbles, okay? These tiny little specks across the whole field, that's what you're looking for, but you're not going to be able to tell the shape until you magnify it more. So don't go focus on something like these big spots. What you're going to see are tiny little specks. Then on the 10X, it's still tiny. These could even be smaller bubbles. I wouldn't focus on those. Do y'all see what I'm talking about? These tiny little spots? Okay, 40X is what you're gonna look at. Here, bubbles, air bubbles. Okay, these are these little tiny specks, and it's gonna be a lot easier to see under your scope, is what you're gonna look at. You're gonna look at them on the 40X, and if you look closely, you're gonna see them moving. You can see them budding sometimes, so if you just watch it, they might move and start budding off. Now, I don't want to say they're moving because they're modal, but it is just a liquid sample, so that liquid could be moving around. Anyways, watch closely and you'll see them start to bud, which is kind of cool. It is alive. Kind of neat, I think. So this is a really good picture. This should be kind of more like what it looks like for y'all today. Okay? For this one, you might need to adjust your diaphragm a little bit. Do y'all remember where that diaphragm lever is? Where it says 100, and then you can move it left and right. It should always be to the left. For the stains today, you might want to adjust it a little bit. Any questions on the wet mount? Okay, the last one is our capsule stain. So this one's pretty cool. The background is stained already. You guys already did it, right? It was stained what color? It's going to be black. So this picture, it's pink. It's going to be black when you guys look at it. And then we're going to stain it with crystal violet, which will stain what? What's it going to stain, though? The bacteria. So your bacteria will turn purple. Do y'all see on here around the bacteria where there's no stain? That's the capsule. That capsule has a neutral charge. So you can't stain it with a basic stain or an anionic stain or acidic stain, which is kind of cool. So any white that you see around the cells is the capsule. So you've heard about the capsule in lecture, I'm sure, right? And what it does, today you're going to see it. So it's really there. It's not just all talk. Here's a picture of the capsule stain, done a little bit differently. I don't love that picture. You do see the capsule stain in the clinical setting, not necessarily, I will say, doing it this way that we're doing in lab today. However, if you guys remember, there's a lot of junk in patient specimens. Do you remember me talking about that with Giardia? In the background, there's a lot of junk because normally it's a stool sample. This one is a sputum sample. So have you guys ever coughed up that nasty stuff and wanted to know what was growing in it? Probably not, but I always do if it's me. But have any of you guys ever had Katie White for anatomy? Yeah, she's the type of person that's like, oh, let's culture it and see what it is. Like, no thanks, I don't want it in here. This is a sputum sample. So in the background, they just stained this patient's sample. And it's not necessarily a capsule stain like what you guys are doing, but everywhere there is a capsule, it didn't stain. So you got that junk in the background, which could be like mucus or it's probably mucus. And then you also have the bacterial cells. So like right here is a bacterial cell, and you see the white around it, that's a capsule. There are a couple different organisms that you typically see in a sputum sample that are causing illness that do have a capsule. So that's good information for that physician to say this sputum sample has an organism that has a capsule. So you don't know exactly what that organism is just by looking at it here, but by knowing it has a capsule, that gives the physician a lot of information because not all bacteria has a capsule, right? So that narrows it down a lot. This is another one. They never look this good, I'll be honest. But that is a patient specimen. Yours today is going to look like this. Your background will be black. Your cells are going to be purple. So it'll probably look a little prettier than this. And then the capsules are white. So wherever you see white is going to be the capsule. Any questions on those three stains so far? Okay. So when you're looking at your stains, remember, I just want to give you a quick little reminder. When you're putting the slide on the microscope, make sure that your light is shining right over where you see color on the slide. So if you're looking at it and you guys have your circle, you know where the bacteria should be in your circle for the simple stain. Make sure you find where there's some color on your slide. You should be able to see it on your slide. If you don't, ask me and I'll look at it. But you should be able to see some color. Make sure this area right here where you see some faint color is the area that you put underneath the light on your microscope. Okay, so real quick, I want you guys to tell me which objective do we start with when we're looking at a microscope slide? Scanning the 4X. knobs do we use on the 4X? You can use the course adjustment, that big knob on the 4X to focus it. I suggest you move the stage all the way up before you even look through the microscope, and then look through the microscope and adjust that course adjustment down. That's the big knob. After the 4X, can you use the big knob again? No. Please, 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 please don't. If you ever lose focus on your slide today, let me know. If you lose it on the 10X, you can go back to the 4X and start over. If you lose it on the 40, you can go back to the 10 and the 4X and start over if you need to. But you can only use the course adjustment on the 4X. Here's the problem. When you're using oil, you're going to focus two of these under oil, your simple stain and your capsule stain. When you're using oil, if you lose your specimen on the 100X, then you have to wipe the oil off your slide and wipe oil off your lens before you can go back and start over at the 4X. However, when you wipe oil off of your slide in this lab, it will take your specimen with it. Okay, so if you lose it on the 100X, ask me before you clean your slide off, because you're gonna wipe your specimen off and then you definitely won't see anything. Okay, I really want y'all to see it. Okay, so let's go over these, how you're gonna stain the rest. Let's look at your pre-labs. Go ahead and get that out. Okay, so you guys already prepped the slides, right? Except for your negative. You guys should only need this sheet out today, and your lab manual can be put up. This is really important. I want you to go in this exact order. Okay? Do your simple stain, do your capsule stain, do your negative stain. When you look at your organisms under the microscope, you have to go in the exact opposite order. You have to look at your negative stain first, okay? I actually don't care if you do capsule or simple, second or third, whatever. Look at your negative stain first. So you're gonna make your negative stain last and you're gonna look at it first. The reason for that is because it can dry out really quickly. You don't want this one to be dry when you're looking at it. Okay, so we're gonna make it last, make them in this order, and then we're gonna view your negative stain first, and you're gonna show it to me. Okay, that's the one that you view under the 40X only. So that's in your lab manual, but you can write it there too. So you're gonna view that one under 40, this one under 100, this one under 100. So do you need oil on your negative stain? No. Please no. Okay, so we are air drying our simple stain, right? We're air drying our capsule stain. Are we air drying our negative stain right now? No. Heat fixing. Okay, this is something I haven't talked about yet. Heat fixing is done to make sure that your bacteria is sticking to your slide. Okay, so heat fixing has two functions. You might want to write this down. It makes the bacteria stick to the slide, and it also kills the bacteria. It makes the organism stick or adhere to the slide. And then it also kills the organism. I mentioned that the wet mount is viewed or used to view live organisms. So are we going to heat fix the wet mount? No, but we're going to heat fix the other two. There is a trick to this, to knowing if you have to heat fix. So we're going to heat fix our simple and we're going to heat fix our capsule, but we're not going to heat fix our wet mount. If there is ever a rinsing step, you have to heat fix it. So after you learn all the different stains, because it's not just these three today, you're going to need to know all of the steps to every single stain. Heat fixing, if there's a rinsing step, it has to be heat fixed. Most of them have a rinsing step, so most of them will have to be heat fixed. But you will need to know which ones. There is no rinsing step for our negative stain, so we don't heat fix it. That's why there's this question here, is the stain going to be rinsed? No. Okay. So we're going to go through these heat fixing. Actually, I'll just go through all three of them with you and how we're going to do them. And as we go through them, you can keep filling this out if you don't have it filled out already. For your simple stain, you're letting it air dry. You have your slide. You're going to get a clothespin once it's completely dry. The clothespins are in the back there. You're going to need one per person. You might need two if you want to. You might be more comfortable. With your micro incinerators, go ahead and switch those on while we're talking about them so they can heat up. You're going to heat fix it, but only when it's completely dry. So if it's wet at all, you can't heat fix it yet. How you're going to heat fix it, your organism is going to be away from the micro incinerator, and you're going to run the back of the slide by the opening of the micro incinerator. Okay, so like on this one, if I were to heat fix it, say my organism is facing you guys and the bottom of the slide is towards me, we just run it across 3 times and that heat fixes it. That's it. Okay, just make sure you get close to the opening so that heat is actually exposing the slide or the bacteria to it. Does that make sense? What is that going to do? Kill the bacteria and makes it stick to the slide. Yep. Any questions on that? If you face the bacteria towards the heat, that could distort it more. So let's avoid that. If you do this before it's completely dry, it could be too much for the bacteria, and it will distort it for sure if it stays on the slide. Sometimes it makes it just pop off. So I don't want it to just pop off. You won't see it because it's so small. But then when you look at your slide, there's going to be nothing on it. Okay? So we have it heat fixed. What's our next step? Does it say? Staining it. Okay. You guys have these pans. Okay, these are lasagna pans. But we're going to use them as staining pans. These over here, there are only 3 because I have one of y'all's, will be for these 2 tables. Those 4 on this rack will be for the middle 2 tables. And the back 2 tables, y'all will use the 4 back there. These stains cannot go down the drain. When you're finished with this pan, we're not gonna, these are brand new, so they'll only look like this today and they'll never look like this again. And that's okay. These are gonna get messy. We're gonna pour out the stain after we use them, and I'll show you how to do that. But at that point, you're not gonna rinse it out or anything like that. So the way you see them stored right now is how I want them stored later. So if you put them upside down, that stain is gonna drip down, okay? Because it's not gonna be perfectly dry when you put them back up. Does that make sense? Okay. So you have these 2 little slide holders, is what I like to call them. They're in the drawer underneath the clothespins. So from here on out, you know that's where they are. If you need one, you can go get them. You're gonna put 2 of these long ways in your tray. Okay, so one of these per person, and know which one is yours. You're gonna set it down. You'll use your clothespin to transfer the slide anywhere at this point. You're gonna put your slide onto one of these while it's laying down, and this is just gonna hold it up for you. So use your clothespin and set it on there. It's just gonna hold it up for you so that you can put liquid on it and it'll fall down. Okay, so it's gonna be setting there, and you have to put stain on it. So you have stain in your trays. They're in the boxes. So the first stain you're gonna use is methylene blue. So you should be able to grab that one out. When I say to stain something, I'm gonna borrow y'all's for a second. I mean flood the slide, okay? Like, don't just put a drop on the slide. Cover the entire slide. I'll put it up here and see if we can tell. If I were to stain this slide, it's kind of hard to see. You definitely want to wear gloves when you're using these stains. And be careful not to get it on your hands or clothes. If it does get on you, it's not a huge deal, but it's gonna be there for a while. If you get it on your clothes, it's gonna be there forever. I spilled some of this on myself on my hand one time, and it was there for like 2 weeks. So anyways, so your slide is gonna look like this. Can y'all see it? Okay. I don't know if that's better. And then you're gonna take your bottle, and you can hold your methylene blue over the tray, and you're gonna flood the slide. How long is it gonna be on there?
1 minute.
1 minute. Okay, so that's what I mean by flood the slide. Methylene blue is over the whole slide. You don't need it to go over the frosted end, but if it happens to, that's fine. You're gonna wait 1 minute. So I'll have a clock up here for you to count or time your stains. And then what are you gonna do? You're gonna rinse it with water. So what does that mean? We have bottles of water. They're over there. 2 per table should be enough. And you're gonna take your clothespin. After a minute, you're gonna pick up your slide, and it doesn't really matter which side that you pick it up on as long as you're not picking it up from the middle where your organism is. Y'all with me on that? Okay, you're gonna hold it at an angle. So I'm holding it so that all the stain is dripping down. And then you're gonna spray water on it until the water coming off the bottom of the slide is not blue anymore. And you may need to rinse the bottom of the slide too. But that looks good. Okay, so it's rinsed. If I were you, I would have a paper towel out just to set this down. So set it down on a paper towel and work on your next slide. Yeah.
And this won't rinse off the organism because it's heat fixed.
Exactly. So there's a rinsing step, which is why we heat fix it. If we didn't heat fix it and we rinsed it with water, the organism would just rinse right off with the water. Okay? Any questions about your simple stain? Okay, you guys have bibulous paper in your trays, which is a really fancy way of saying absorbent paper. You can just take this bibulous paper and open it up to a random page, put your slide in there, press down. Don't press down super hard, just kind of pat it a little bit. That's going to take some of the water off, and then set it aside. Don't rip the pages out of the books or anything like that. That's all it's there for, to take some of the water off. You don't have to wait for it to completely dry before you view it under the scope. However, I do want you to set up all 3 slides before you get your scopes out. Okay, so your simple stain is finished. What's your next stain? Capsule stain. What are the steps of the capsule stain? You're gonna heat fix it. So the side with the India ink and bacteria is gonna be facing away from the opening to the micro incinerator. and you're going to run it by 3 times. Then you're going to flood the slide with Crystal Violet. How long? Okay. Same thing with the bibulous paper here. Okay, for your negative stain, we haven't really talked about this one. You have to start this one with your slide that you haven't done anything with yet. What I want you to do is you're going to make this one last, if you remember, because it's going to dry out fast. You're going to put a drop of India ink down in the middle of the slide, and you're going to put a drop of water in the middle of the slide. If I were you, again, I would just use the dropper, but you can use a loop full of water if you want to do that. And then you need to micro-incinerate your loop. And what organism are we going to use for this one? Saccharomyces cerevisiae. It's fun to say if you want to practice. It makes me think of, have you guys seen, well, you probably haven't. There's this like microbiology cartoon where it jokes about the terms being like a Harry Potter spell or whatever. I don't know. I think of it now every time I say Saccharomyces. That one in particular, I don't know why. Anyways, you're going to use that one. You're just going to tap down on one of the colonies. It's over here as well. And mix it into that India ink and water. Then you're going to put your loop back in the micro incinerator. And you're going to take a cover slip. So you guys have cover slips in your trays. And you're going to cover that liquid with a cover slip. Make sure you're wearing gloves because it will probably get on your gloves. Yeah.
So the water, India ink, we're putting in the organism, and then we're covering it.
And then the cover slip. Yep. And this is the only one that you need a cover slip for. Okay. You're looking at this one on the 40, so don't go to oil with the cover slip on it. You should be able to see everything just fine on the 40. How do you guys feel about this? Okay. Okay, I want you guys to clean the negative slide first before you do anything with the staining. Go ahead and clean it so you don't forget. Use the grease lightning on it. You're going to need to wear goggles and you're going to need to wear gloves for everything. Yeah.
So after we stain everything and get all these prepared, you want us to look at the negative one first and then do the...
Yes. I want you to show me all of the slides. Okay, I want you to show me your scope when you're finished looking at all of the slides. We have extra credit at the end of lab today, so let me go over this real quick if you don't want the extra credit opportunity. For next week, study lab 8, read over lab 9, and then there's a pre-lab for lab 9 in the appendix. Everyone got me on that? There's a pre-lab for lab 9 in the appendix of your lab manuals that you need to have done for next week. Okay, but once everybody is finished looking at their slides, I'm going to give you guys extra credit. The other important thing, your micro incinerators cannot be on the table at the same time as the microscopes. Okay, that's really important. So when you're finished with them, put them on the side counter somewhere or something like that and let them cool before you get your microscopes out. So if 3 of you guys are finished making your stains and 1 of you isn't, you all have to wait until everybody's done with the stains and the micro incinerators for you to get your scopes out. Okay, any questions? Yeah.
What are we looking for in the results section of each?
Okay, in the results section, you'll just write what you see under the microscope.
Okay, just describe it.
In your words, yeah. You guys are going to get to keep these. I'm not taking them up, so it's just for your information.
Okay.
Okay, let me know if you have any questions. While you guys are checking everything and cleaning your tables, I'm going to go over this extra credit, okay? And then I'm going to pass it out. Now, I will say we're going to go over this like a few of the questions, but not all of them. But I want you to answer all of them on your paper. So this one is about personal freedom versus public health.